monohybrid cross. So a monohybrid is a one trait cross. So this is the normal Punnett square that you would see if we were in school. So you would see these drawn on the board, and then we would be able to write into them, and I would just be like writing all the letters by hand on the board, and then we would like fill them out together like this. Now, unfortunately, this looks kind of ugly, okay? And it was really hard for us to input these into Google Docs. So therefore, you're going to see a different Punnett square on your Google Doc papers. So on your Google Docs, you're going to see a Punnett square that looks like this. It's going to be much easier for you to type your letters into, since some of you do not have the ability to write on your documents. So this is our first practice problem. It's an easier problem because you're given the genotypes of the two types of pea plants. So you're given the parents' genotypes. Much easier than if you had to figure it out on your own. So the first thing that you should always do in a Punnett square problem is that you should read what is given. So they say a green pea plant, which has the genotype of capital G, lowercase g, is crossed with a yellow pea plant, which has the genotype lowercase g, lowercase g. So then the first thing you're going to do is you're going to then put the genotypes into the Punnett square. So at this top of these two boxes at the top here, this is where the first parent's genotype is going to go. So I'm just going to type it in there. So we have capital G, then lowercase g, and then the other parent's going to go on the side here. So lowercase g, then lowercase g. Okay. So then what you're going to do is you're going to put them together. So what this really is showing you is that this capital G would be in like the sperm cell, the one gamete, and this lowercase g would be in the other gamete. So this would be in like the egg. So when they come together in fertilization, this is then what the child or offspring is going to get. So you just bring the one letter down and then bring the other letter over. Now you always want to put the capitals before the lowercases to show dominance. So I'm just going to type in the capital G and then lowercase g. And then over here would be two lowercase g's. And then you have capital G again, lowercase g, and then the two lowercase g's. Not sure why it's like not all showing you the same font, but you guys can see. So this is what the children then would have. So this is the potential offspring. So then what you're going to do is you're going to then put in the genotypic ratio. Now when you're writing your ratios, you always put the number of homozygous dominant first, then heterozygous, and then recessive. So in this case, we have no homozygous dominant. So I'm just going to put 0. Remember, the genotype or the letter combination. Then here we would have 2 heterozygous forms, so big G, little g. And then here you would have 2 lowercase g's, lowercase g's. Okay, so that would be the ratio. So sometimes you don't even need to put the big G in. It could just be 2, so it'll be 2 to 2. So 2 capital G, lowercase g to 2 lowercase g's, lowercase g's. Now, the phenotypic ratio, remember, phenotype means what they actually look like, so the appearance. So in this case, you always put the dominant appearance first. So there would be 2 green pea plants. So that would go with the capital G, lowercase g genotype to 2, then yellow pea plants. So that would be everything you needed for this Punnett square problem. So you would actually read it, put in the parent's genotype and the Punnett square. So there they are. And then this would be the potential offspring. And then you figure out the genotypic ratio and phenotypic ratio. All right, so our next example is a little bit more difficult than the first one we just did because they don't give you the genotypes of the parents. You have to read what is given and then figure out the genotypes yourself. So in this case, they say red color, capital R, and white color, lowercase r. So that means red is going to dominate over white. So a flower who is homozygous for red color is crossed with a heterozygous red flower. So I'm just going to underline that. So we have homozygous red and then heterozygous red. So then I'm going to type in the genotypes here. So we have the first red flower is homozygous for red. So homozygous means same. So that would be capital R, capital R. Now, heterozygous means different. We know it's still red though, so that means it has to have at least one capital letter. So that's going to be capital R, lowercase r. So I have the two parents' genotypes. It's very important that you get those correct, because if you don't get those correct, then the rest of the problem is going to be all screwed up. Then I'm going to put the genotypes for each parent onto my Punnett square. So I got my first parent, and then down here is my second parent. Okay, then I'm going to put them together just like fertilization would. Okay, ta-da, our offspring's filled in here. So then what you're going to do is you're going to use this information for the potential offspring to answer the genotypic, phenotypic, and then probability. So with the genotypic ratio, again, you always write homozygous dominant first. So in this case, we have two capital R, capital R's. Then we have the heterozygous would be two capital R's, lowercase r's. And then here would be 0 lowercase r's, lowercase r's. Then the phenotypic ratio, remember to have a dominant phenotype, you either are homozygous dominant or heterozygous. Because even if you have one capital letter, you show the dominant phenotype because it overpowers the recessive allele. So in this case, there would be 4 red flowers to 0 white. So then it says, what is the probability of a red flower? So these two plants, these two red plants were crossed together, the potential offspring would then be all red. So it says, what's the probability? You would say 4 out of 4, or you can say 100%. So that's how you would do one of these problems. Here's our next practice problem. So it says, in tomatoes, red fruit color, capital R, is dominant to yellow. Suppose a tomato plant that's homozygous for red is crossed with one homozygous for yellow. Determine the genotypic and phenotypic ratio of A, the F1 generation, and B, the F2 generation. So really this is asking you to do two different Punnett squares. So we're going to do the F1 generation. So to figure out the F1, you're just going to cross the two different parents that they're giving you. So again, I'm just going to highlight the information here or underline it. So we have homozygous red, and we're crossing it with homozygous yellow. So those would be the parents. So this would be our first genotypes we're going to write down. All right, so here's the first one. It's going to be homozygous for red. And then over here, homozygous yellow. It's going to be 2 lowercase r's. Then I'm going to put them on my Punnett square. And then I'm going to put them together. Okay, so you can see here, this would be the genotypes of the offspring. So then I'm going to write in my genotypic ratio. So again, it's homozygous dominant first. So we have none of those. All right, then we have 4 capital R, lowercase r's. And then we have 0 lowercase r's. So then the phenotypic ratio for this F1 generation would be 4 red to 0 yellow. Okay, so that's the first part of the problem. So the second part of the problem is going to ask you to find the F2 generation. So if you can remember back to Mendel's experiments, when he did the F1 generation, he found that they were all hybrids or they were heterozygous, and that's what we found. We have found 4 capital R, lowercase r's. To find the F2 generation, what you're going to do, just like Mendel did, he's going to take 2 of these plants and he's now going to cross. So you guys can see for the F2 generation, I've taken 2 of the plants from the F1. So remember, they were heterozygous. So here's the genotypes of the 2 plants. Now you're going to put them into the Punnett square. and then you're going to fill in the Punnett square. All right, so now that the Punnett square is filled in, we're going to go up here and write in the genotypic ratio. So in this case, we do have 1 capital R, capital R. We then have 2 capital R, lowercase r's, and then 1 lowercase, lowercase. So to find the phenotypic ratio, you just have to go back and remember that the way that you have a dominant phenotype is if you have a homozygous dominant or heterozygous, because heterozygous is still a dominant phenotype, because if you have at least 1 capital letter representing dominance, it masks the lowercase letter, which is representing recessive. So in this case, you would have 3 red plants to then 1 yellow. So in the F2 generation, that recessive trait does show up again. So I hope this little tutorial helps you guys. I'm going to keep playing with it, and hopefully I can make it a little bit smoother and make sure the letters are all the same size and things like that. But if you have any questions, please email me.